Like to introduce Bjorn back, who presented yesterday, to talk about the T cell class 2 tools, binding, processing, and immunogenicity on the classic side. Thanks, Bjorn.
I should be ready to go. Okay, so I'll talk about the MHC class 2 restricted T cell epitope prediction methods that we have, and I'll in this process also talk about the TEPI tool um, approach to making predictions, which actually covers class 1 as well. So this is going to be all on the classic IDP side. And I mean, looking at what Jay presents and Dan, I really look forward once we migrate things over. We have been neglecting new updates on the site a bit in the anticipation that we're going to move things over to the next generation site, hopefully within the next few months. So I want to start off with the biological background a little bit on what's the difference here between MHC class 1 and class 2 restricted T cell epitopes. So class 2 restricted or bound epitopes are recognized typically by CD4 T cells. So they have a CD4 co-receptor. They are typically derived from extracellular proteins that are taken up, processed in the endosome, lysosome, and bound by MHC class 2 molecules. They are trimmed on the sides, but as you can see here, there's kind of like this core part that is binding, and that's then presented on the cell surface to a CD4 T cells. And these are typically, this is in contrast to MHC class 1 molecules. These are not found on every cell. These are only found on professional antigen presenting cells. Yeah. And again, like the key difference here is also that these are typically from extracellular proteins rather than internal proteins. So this means that they are typically presenting um, antigens that are derived from extracellular environments, such as extracellular bacteria or allergens, secreted proteins, debris from dead cells, and that's what makes them also intrinsically different from the kinds of things that class 1 molecules present. The CD4 T cells, when they recognize something, can both have direct effector functions. They are cytotoxic CD4 T cells and things, but they are more classically known as helper T cells that actually help B cells that produce antibodies to make a more efficient response. And they can also provide help for CD8 cells and kind of both trigger them to kill more or reversely to kind of like stop an immune response if these are regulatory CD4 T cells. So the MHC class 2 molecules are different primarily, as I mentioned in the last slide here, that they bind this core part of a peptide, and then the sides are open and they overhang. Genetically, they are made up by two chains, an alpha and a beta chain. And in the case of, so there's three different loci, DR, DP, and DQ, and they come both with like an alpha chain locus and a beta chain locus. And in the case of DP and DQ, both the alpha chain and the beta chain is polymorphic. So essentially, you have like um, theoretically combinatorial possibilities of having all the alpha chains from DP can pair with all the beta chains from DP, and then the same for DQ. And then for DR, you only have the beta chain being polymorphic. So typically, when you refer to a DR class 2 allele, you say something like this is DRDP1 or 101, and you're assuming because the DR alpha chain is monomorphic that this is there is always a DR alpha chain bound to it, but this will always be essentially DR A1 or 101 with some minor exceptions, but that doesn't really matter. And then we have the DRB3, 4, 5 locus, which is interestingly quite often overlooked, although this is one of the most, so this is essentially like a locus that also pairs with the DR alpha chain. And these are not so polymorphic like the DRB1 locus. So actually, these are often like very frequently found in the human population, each single allele of them. So that is the MHC class 2 locus. Then the binding groove, as I mentioned, is open and can accommodate peptides pretty much from 13 to 25 amino acids. Once you cut out to be less than 13 residues long, typically the peptide will no longer bind to class 2. It needs some kind of overhang. And then you have the 9 amino acid binding core, which really makes the direct contact. And as you can see here in this example, you can have, because really just the core is strictly specific in terms of the amino acid composition for binding, you can have peptides that look quite different and can all bind. And so this is one of the examples that makes this a little bit more challenging than class 1, analyzing class 1 binding or illusion patterns, because you kind of have to align these first to see how they have a same core. And then obviously there's multiple ways of aligning things. So as I mentioned, the tools are on the classic site. So if you go here to the IDB analysis resource and then go for MHC class 2 binding, you're getting a page that should look very familiar at this point. This is the typical input page that we have for most of the classic tools. And essentially, it asks for a sequence input. And then for class 2, it gives you the binding methods that are available. And so you select here. And as before, we are using the results from these benchmarking runs that are where the data that is newly published in the IDB is used to evaluate how good the different tools are. And based on that, we've been updating the predictions that are being recommended by the IDB. So the next thing where a class 2 is different is, as I mentioned, we have this massive, we have the different loci, and then for each loci, we have the different combinations. So just having a list of all possible HLA class 2 alleles, and let alone than other species, would be tens of thousands, probably much more. You can't just list them all. So instead, we have this drop down where you first have to choose your species and locus. So say you choose DQ, then you can select the alleles here from a drop down. And these alleles that are shown to you are one where there has been some kind of experimental evidence that the alpha and beta chain pairing actually is made in nature. So you can also select things here if you click this check marks independently, and then you're choosing first the alpha chain, then the beta chain. You can also have the full HLA reference set. This is similar to what Jay explained for class 1. These are alleles that have been frequently found in an attempt to, I think it's like 23, that are frequently found in the human population. And essentially, if you include all of that, you're going to be covering lots of different specificities that are common. I mean, many of these rare combinations that you have are going to be found in like 1 in 10 million people. So it's unless you have a specific reason, you probably would not want to study those. Then we have this other specialty for class 2, which is the 7 allele set, which I'm going to explain for a second. As I mentioned, we have this issue that for peptides, you have different registers in which they can bind. So, and then you also have, in contrast to class 1, you actually have a lot of overlap in the binding specificities between these different molecules. And then they can end up with, you have like a 15-mer peptide, you can have different alleles binding in different parts of that 15-mer. So essentially, to evaluate a given peptide, that's in 15-mer is what we typically use, you have to evaluate all the different possible cores in there, and for all the different possible alleles that they can have. And you might have a peptide then is much better than others for likely T cell recognition, just because multiple different alleles can bind it in different or the same site. So we've been trying to come up with an overall prediction method that best predicts from a given, just the peptide alone, is there going to be likely going to be a CD4 T cell response or not? We used sets of peptide data sets that were tested systematically for immune responses in T cell assays, and then asked, can we re-identify them, or could we have predicted them? That's the way to say it. Could we have predicted them based on MHC class 2 binding restrictions? And we ultimately found, these are just the data sets, that a set of 7 alleles, if you predict for these and then take the median affinity of those, then you end up with like a good measure. We had the best measure that we could find to predict the likelihood of the peptide to be immunogenic. And when you then look at these alleles, these are actually very, very frequently found in the human population, each of those. So to some extent, these are like different specificities that are overall making it likely that a given peptide is being recognized by something. Now, this is very different from a prediction approach where you know you only care about whatever, one patient, and that patient-specific expressed alleles or so. This is an approach that is useful if you're trying to design a peptide set that is going to be likely to be recognized in a broader outbred human population. So then going on, you have to select the peptide length. And again, like we use 50 mers. That is pretty much because you have if you have a given 9 mer, you have 3 on the side. It's a nice, but we found that if we use overlapping 50 mers, we kind of like pick up every epitope that you can find in a given protein sequence. And actually, I'm going to show that pretty much next. So now you're scanning this input sequence with this recommended method for, in this case, just one HLA-DQ allele that I selected, and for 50 mers, then you're getting this kind of results back as before. Here again, the explanation is that a high score in the case of this method is a good binder, so you can sort these, and this is the highest scoring peptide, and it'll tell you not just what the peptide was that is meant, but also where the core sequence, the 9-mer sequence essentially here, that is expected to make direct contact to the HLA molecule. So we have some guidelines for selecting these binders. So we have originally said, like 10 years ago, we would have definitely said class 2 predictions are much, much worse than class 1. But as we have seen over time using our automated benchmarks and other publications, these AUC values have gotten better and better. So the predictive power of these methods has really improved over time. Originally, we had something said that like a percentile rank of less than 10. Again, these percentile ranks are if you take a score like this one here, you have these scores, and then they are applied to like 100,000 random peptides. Oh my God, my battery's not low. That is not good. That is not good. That shouldn't happen. Perfect. I blame Trump. Okay. Uh, I hope everything is still okay. Okay, these percentile ranks are essentially then calculated by taking the score that was found and applying this to 100,000 peptides and then ranking the highest to the lowest score. So this is in the top 1.3% of scores found. So yes, so this is what I was trying to say. That sorry, the power really threw me off. Um, Being in the top 10 percentile rank was what we originally recommended as being a reasonably good binder, and an IC50 value of less than 1,000 nanomolar. But we have seen over and over now that these are probably too loose, the criteria, and we can probably tighten them up and be more stringent here in the selection method. But we still haven't. We want to be conservative when we make changes to our recommendations, and we still haven't found a good enough data set that makes us really confident in changing these recommendations going forward. Now, I want to point out another thing that is tricky with the class 2 binding predictions. So the current approach that I showed you with this tool, it takes the long amino acid sequence, splits them up in all possible 15-mer residues, and then makes the predictions for them. So what then happens is, as you can see here, you have a peptide position 527, then 528, 526. These are all overlapping peptides that are ranked at the highest value here. And essentially, if you were to make all of them, they are essentially pretty much the same peptide that you would be testing. So that would be a waste of money. So what you can do is you can take your predictions and just remove the peptides that are overlapping. You sort them from best to worst, start with the best, take it, and then remove the ones that are overlapping with the one that you've already selected. That essentially summarizes, I think, what I have here for the classical prediction method. Then switching to TEPI-tool, which automates some of the things that I showed you before that are kind of challenging, such as in the 7-allele method that I showed you, you would have to manually do the median value for all the different alleles. for the post-processing, you would have to do that somehow manually later as well. So the TEPI tool was our original approach, how to combine and kind of like pipe different steps in a prediction method. And to a large extent, this is going to be replaced by the next generation tools that Jay showed. But for class 2, as they aren't available yet, this is still highly relevant. So for TEPI tool, you start with a protein sequence. This is the same start for class 1 or class 2. You choose your species, host species, and your allele. So here you switch between class 1 or class 2. I'll briefly touch on class 1 to also show like the history kind of TEPI tool. So then it asks you which class 1 alleles do you want to pick. Here I'm giving as an example, you can pick 3 just from a list, but you can also do, like Jay showed also for the next gen tools, you can use panels of say the 27 most common alleles. And you get like an echo back of what you have selected so far. You get asked how you're going to deal with duplicates in your data sets, which can occur. Specifically, if you have similar peptide sequences, like what Dan showed in a much more fancy way. Here you can just remove essentially identical peptides that might come up in your predictions. And you can perform a conservancy analysis. This is again similar to what we can do with other tools. But here it essentially asks like if you had multiple peptide protein sequence as an input, such as the different dengue sequences that Dan had, you can ask that you only want to have peptides that are found in at least 2 of these input sequences. Then after you've made those selections, you get asked which prediction tool to use and can choose here again from the kinds of methods that were available here. There's more available now on the next gen site already. So for class 1, this is really kind of outdated. And then you can also do your prediction selection of the ultimate peptides based on different approaches, like using MHC binding thresholds, predicted percentile ranks, and so on. There is help available from TEPI tool that explains the selection of these cutoffs. And again, what Jay showed for next gen is covering much of these. Then you can also here in TEPI tool choose the specific allele-specific cutoffs, and then review and submit your data to the tool, and then you get an email back kind of when this is done. In contrast to like the pipelines and the next generation tools, you cannot persist this. You cannot couple this with other methods that we are new developing in this wizard. What we found and the reason why we moved away from it is kind of we are adding more steps to it, becomes increasingly complex and makes it hard for us to maintain this approach, which again is why we switched to the next gen approach. So for class 2, there's a couple of differences. One is how we deal with overlapping peptides, as I showed you before, which is an issue for class 2, but not for class 1. So we can do approaches like having peptides initially scanned. I chose 15 MERs before here, and then essentially looking for peptides that are overlapping at most by 10 residues. That'll essentially give you, when you choose 15 MERs, that are 15 MERs overlapping by 10, so like 1 through 15, 6 through 20, 11 through 25, and so on. And that tends to be like a very practical approach of looping sums in, and then only those will be run through the prediction methods. This again is for the prediction methods available. This is again the same selection as before, and then review and submit these predictions. The other thing, as I promised, there are alternative methods here that are again specific for class 2 in this step 5. One is that you can select based not just on what I showed you for class 1, but in addition, you can select for number of alleles binding. Because again, like this is in a given peptide, you might have different alleles binding in different registers, essentially. And you can use that as a prediction threshold to pick peptides that you think should likely cover a broader set of alleles. Similar for the 7 allele method here, you can predict using pre-selected choose like to predict using pre-selected alleles, sets, and method. That exists for class 1. And for class 2, we also have the 7 allele method, which then specifically selects these 7 alleles. And then we can use in step 5 the selection method promiscuity based on 7 allele method, which is that score that I mentioned before of the median of those 7. So um, I want to switch now to MHC class 2 processing predictions. They are found here on the side, MHC2NP. This tool uses MHC2 ligand elution data to predict essentially the generation of the peptides in this as they are processed in the lysosome before they bind MHC class 2 molecules. And that's what's shown here. Again, you have the protein being taken up, being processed here originally, and then trimmed as it's bound to MHC class 2. So we knew this would happen. And you have essentially, there's different ideas of how exactly it works, but essentially you have probably the core bound already to the MHC class 2 molecule as further processing can happen. And then ultimately this is transported to the cell surface when that is done. We're using essentially, if you go back here, data from peptides that are eluted from the cell surface to get at like what was here, where was this cleaved from? So we are taking naturally processed peptide data for MHC class 2 molecules, and we're doing a cleavage motif analysis, essentially at what are the residues that are preceding the termini of these peptides, and that are then directly at the termini of this peptide, and looking for a statistical signal that indicates an enrichment of certain cleavage motifs. So as I'll show you in a second, the ligand prediction, so predicting which peptides are actually on the surface, is markedly improved when combining these kind of cleavage motifs with binding alone. So that works great. What was super frustrating for us, and I'm going to say this right now, so before you get excited, is that this approach did not end up improving T-cell epitope predictions at all. And well, I can show you the argument for that, why that is later. So this is how the tool works, and there's reason to want to know what ligands are on the cell surface, independent from epitope prediction. So this is still a useful tool. So you can enter your protein sequence as before. You submit this, and then you end up with these data here is like an echo essentially of the proteins that were input, and then there's the top 5 peptides here that are given. You can also just see the complete results. We're not showing this by default because then it takes so long. And this is just a TSV file. And then here essentially you're getting the peptide sequence echoed. You get the N-terminal motif, which is, you see, is the SVP. So there's an S preceding the peptide in the protein sequence. And pKa, so it's like the PK in the peptide and the A post. We found that these tend to be the minimal sequence region that really is conducing the likelihood of something to be cleaved. And then we have cleavage probability score and ranks that I used to get to these. Yeah, when we have these cleavage scores and we predict natural eluted ligands with them, we can combine it with a binding score. Here's just a simple linear combination of the two with different weighings. If you use only binding or if you use only cleavage on like a blind data set, you'll find that in this combination, you're getting a clearly improved score by combining both. That is published in this paper. Now, and it was actually interesting and fun that Morten's group in Denmark at the same time had a similar study, and we ended up exchanging results and finding the exact same thing. And we were both struggling to find out why this doesn't actually help with immunogenicity predictions. And so the reason is ultimately that these cleavage motives are very, very broad. So essentially, if you have like five possible cleavage sites, one of them is going to be good enough. So, and given that the MHC class 2 molecule can bind very long regions, essentially lack of a cleavage site is never going to hurt the ability to present a binding core that has high affinity. It'll just be longer or shorter, depending on where the cleavage sites are available. And the part that the T cell recognizes is really primarily overlapping with the MHC binding core. So the length of the overhanging um, regions on the side do not seem to matter that much, and we end up being very good at predicting ligands, but not we don't improve here with this cleavage score predicting T cell epitopes. I hope that made sense. I am going to now move to the final one. I realize I'm probably way ahead. I sped up too much, being scared by my power running out. And I'll talk about CD4 T cell immunogenicity predictions. Um, This is a server that we developed to get at these problems, right? Very often we want to know from a given which peptides in a given protein are really recognized by T cells and causing an immune response, so immunogenic. So, and as we've tried multiple different things here, and the MHC binding approach still has been working great. In the population, the 7 allele method works good. But we thought there must be other features in peptides that make them more immunogenic, some than others. Very similar to what Jay showed for the class 1 peptides, where we found this enrichment of certain residues. So we took a bunch of peptide data sets, proteins from the IADB, for which overlapping peptides were tested for immunogenicity. The reason why we are using overlapping peptides here is we want to have data sets that are not pre-selected for any kind of other predictive selection so that we can evaluate predictions in an unbiased fashion on them. So we had these peptide sets and then trained a neural network just saying these were immunogenic versus not, and then try to see can we generalize from that. So that's the same kind of data set that the 7 allele method was essentially created on. So where we then used these combinations of MHC binding affinity predictions to identify which peptides were immunogenic. So we have now a separate kind of prediction that doesn't take alleles into account at all, but goes straight for trying to predict based on some features, whatever the neural network picks. So we also picked a tetramer data set derived from the IDB, so that's a nice one where we can it's less number of peptides, but where we know for certainty the MHC allele restriction there, and then we can compare which ones were immunogenic or not. And ultimately, we took 57 independent studies from different groups across the world and tried to validate this. And then we implemented the three approaches. One was the 7 allele method. I mentioned the immunogenicity predictions that I showed in the last slide, the network, and then this hybrid approach, which is really the goal here of trying to enhance what we got from the 7 allele method by itself. This is the tool. It's called CD4 EpiScore. This is the input to the tool. It should be very familiar now. And if you then pick the combined method and sort this by percentile rank. No, actually position the protein here, I guess. I don't know. And then you're getting your immunogenicity scores and the affinity predictions for the 7 alleles and the median of those. And then you have your combined score, which is really what is being used here. And this shows how far the improvements went in the combined score, going from an alpha of 0, where it's all 7 allele method, and an alpha of 1, where it's all the immunogenicity score. And you see we do have some improvements here in the range of 0.02. Oops, this is not my day. In the range of 0.02 for the AUC value. And so again, this is all published. The reason why we haven't been pushing this further and why we have not let the tool come out of the lab stage, which it's in right now on the website is that we have not been able to consistently reproduce this kind of improvement using this immunogenicity score in other data sets that we have subsequently worked on. Okay, that is the last thing I had. Now let me summarize. The performance of MHC class binding predictions and ligand dilution predictions has massively improved in the last years, but we have not yet updated the thresholds. As I mentioned, we're trying to be very conservative with that. We are currently recommending the NetMHC2 Pan BA family of tools that are predicting binding affinities for when you want to have really IC50 values kind of as an output, and EL scores if you want to have essentially probabilities of finding the peptide processed and presented. As I mentioned, we have the promiscuous nature of MHC class 2 binding that makes it more challenging or makes it more important to look for kind of overlap of affinities across different alleles and the ability of the same peptide being bound in different registers. All of this leads to peptides that can be broadly reactive. So we have peptides that are easily recognized by 50% or more of the human population in different alleles in different individuals. TePi is our approach here to shortcut some of these post-processing steps and making that done in a tool where, again, we are aiming to transition these to the next generation tools. And as I mentioned before, we are constantly reevaluating these epitope predictions, and all of these recommendations we are making here are subject to change, definitely for class 2 more so than many others. So I'll stop my sharing here. Great, I wasn't even in view. And so we're done way early, which is good because we have a lot of questions. I'll just rearrange my computer for a second, Nina, I'll go off.
No, you take your time. Thank you so much, Bjorn. Yeah, we are running about 15 minutes ahead, which is actually great for the Q&A because it just means we'll add that time to the existing about 20 minutes that we had because there's quite a few coming through. So I'll let Bjorn take a look at the Q&A, and while he does that, I will invite Jay and Dan back to kick us off with the first Q&A for day 2, our extended one. So, Jay, would you like to take the first question?
Sure, yeah. And so we had a lot of questions related to how long we're going to store links for pipelines. And the answer is our goal is to be able to store those indefinitely for now. And we'll reevaluate that as the new next gen site stretches its legs and what that's going to mean for us. But the goal is that they will be indefinite.
I can take the next one. There's a question about how PepMatch performs compared to BLAST and would it report different results? Yeah, so we published a paper on PepMatch last year, and we do a comparison between BLAST and PepMatch, and PepMatch is designed for short sequences search in large sets of proteins or proteomes. And it's more deterministic than BLAST, which is more alignment based. So it is different, but it's more accurate for short sequences.
Thanks, Dan. I'll take the next one. Anonymous NTD asks how negative set was selected for training and testing of NetMHC Pan. Essentially, all the Pan specific methods that are primarily generated by Morten's group in Denmark use the IADB data, both the positive and the negative binding data, and as examples, and then in addition the MHC ligand dilution data, where they use essentially everything that is eluted is considered a positive. For the negative data set, there's different approaches and they have changed over time. I would really go for the specific papers. There were attempts of mismatching, of saying using the same peptides that were positive for one allele as negative for the next, which works in some cases, but not in all. There were other approaches where we then used essentially a random background set for negatives. So for MHC ligand dilution data, the question of what negatives are best to use for training these kinds of networks is kind of an active research question. Thank you.
I could take the next one. So there's a question from Kiran. It's a two-part question. Does the TC1 tool support the generation of wild type and mutated peptides from FASTA sequence? And does it provide predictive values for these peptides in a way similar to the PVAC tool? So no, it will not generate wild type and mutant peptides, but we do have a couple of new tools that you'll hear about tomorrow that are kind of geared towards that workflow. There's the mutated peptide generator, which will take in a VCF file and predict the new epitopes that can be generated. And then there's a peptide variant comparison tool, which will take the wild type and mutant peptides, run them through various binding tools and immunogenicity tools, and then allow you to compare the results. And then the second part of his question is peptide generation in TC1 based on a sliding window approach, or does it use the NetChop algorithm for peptide cleavage prediction? Yes, it's based on a sliding window approach. So it's going to produce peptides of the length that you specify, shifting over by one residue every time. Thanks.
So there was a question, or actually two questions that are very similar. One is on whether you can choose the clustering method for the cluster tool. There are a few methods in the clustering tool, but they're all based on sequence identity and graph theory, not on k nearest neighbors or k-mean clustering. And the other question was just about whether those parameters I mentioned you can use in the current version, and yes, you can. So it's just based on those implemented in the clustering tool as I described, and then if you want to see further details, there's a paper on that as well. But again, not on k nearest neighbors.
The next question I have is from Elise Ishida. How does IDB predict binding of unnatural amino acids to MHCs? That's an easy question because the answer is we don't. So unnatural, definitely not at all. And that would be also really hard. We're always requiring enough data that's going to be available. What there has been significant effort on is to try to look for binding of post-translationally modified residues, such as methylation or phosphorylation of peptides. And that ends up being really hard so far. We have not managed to come up with really great predictions methods. That's why none of them are currently available on the website. But we're totally wanting to do that. Jay?
Yep. So the next question I have is from Mauricio. Will the new generation tools be the default option soon? And so I would say they should be the default option if you're going to do T cell class 1 predictions or any of the methods that are available there. And you can, there's a link to them from the top of the main IDB database page. And then there's also a link when you go to the legacy site that says try our new prediction tool. So I would consider them to be in a production state, and I would direct you to use those tools if they meet your needs.
My next question is, can you use PepMatch with a custom user-provided proteome? Not on the next-gen site, so the data needs to be pre-processed before you can search on it. So we store the data on the back end. So we have like 8 proteomes you can select from, but you can also use the tool separately as a standalone and run your own search. So you can provide your own proteome to pre-process, and there's documentation on that. Link. Let me know if you can't find it, and I'll link it.
Okay, the next question I have is from an anonymous attendee. Some epitopes bind multiple HLA alleles. Sometimes they can overlap with each other. That is, the sequence of one can be sub of the other, and so on. How can I, in this case, be sure which is the one that has potential in downstream analysis? So this is fundamentally different for class 1 and class 2. For class 2, I would just take the longest one that incorporates all of the ones that you have. And that tends to be the most powerful one because there is no really much of a downside to have a little bit of overhang for using them in multiple alleles. For class 1, it's often the opposite, even though you might get a response to a cell, to a peptide that is kind of longer than the optimal one. The optimal one tends to have higher affinity and be much more effective, definitely if you have lower concentration available of it. So there, the best approach is actually to cut them down and titrate them down and see which one is really giving you the response that you needed. That's it, Jay.
Yep. The next question that I have is from Krista McCutcheon. It says, on the next gen T cell prediction tool, I want to make sure I know where the documentation is to understand the biological relevance of the percentile ranking numbers in the table. And so I will paste the link to the documentation in the chat. I'm not sure if I can do that right now, but I'll do that right after this. But the documentation is on, if you go to the NextGen tool site and go to the little help and info drop down in the top right, and then you can actually search the documentation for the word percentile, and you'll find some documentation there. And I'm just taking a look at it right now, and I think we can probably enhance it with maybe some of the content from the slides that I showed. Thanks.
There's another question regarding PepMatch. So PepMatch will eliminate potential self-antigens. I'm not sure exactly what you mean, but the tool will search peptides in proteomes, and you can search like viral proteins in the human proteome for conservation and things like that. So the keyword potential is obviously very key here, but it's not a tool that does anything about predicting actual immunogenicity. So
So the next question I have is from an anonymous attendee. It's yesterday Björn kindly explained that the percentile score rank can correct for peptide length bias, but if the pool consists mostly of 9-mers, then random selection will not help unless weighting was applied upon random selection. So this is actually a really interesting question, and very, very good catch here. So if you are the way we have worked with this is that we are making the predictions in parallel for different lengths, and then essentially have as our negative set the same number for peptides of each length. So that means the neural network has to kind of find out what is the right weighing to give you the right number of peptides of each different length. If you don't do that, and if you train separately, or if you adjust that you have the same number of negatives for 9-mers as you have positives, and the same number of negatives for 10-mers as you have positives, the network has no way of learning that 10-mers are less frequent than 9-mers. So essentially, we are taking the natural length distributions now as our positives, and then using essentially like all the possible 9-mers, 10-mers, 11-mers, whatever, from the underlying data set of protein sequences, and then the network itself will do the weighing. So and then essentially all the ranking we're doing is again like on this universe of negatives. So this is yeah, but actually it took us quite a while to find this out. Also because like when we do our peptide evaluation in the past, we've always done them separate for different lengths. But the real life situations, typically you have a protein sequence and you want to be told what is the length distribution that is the most effective representative of your positives here. Yeah, that's it. Dan? Yeah, no, Jay, sorry.
Yeah, so the next question that I see is from anonymous attendee. It says, hi, Jason, just to confirm, if the peptide binds any of the 27 alleles, it covers 97% of the population. So no, what that allele reference panel is it's a set of 27 alleles. If you have a pool of peptides where one of the peptides in that pool binds to at least one of the alleles. So if all of the alleles are bound by at least one of the peptides in that pool, that is what will cover 97% of the population. Thanks.
There's a question on cluster tool again. Would the cluster tool be used to find potential cross-reactive peptides? Potentially, right? So if you have a sequence identity based on 80% and cluster peptides are very similar together, they are potentially could be cross-reactive. But again, it's based on sequence identity conservation, not on anything regarding reactivity itself.
So next question I have is from Steve Darnell. How is binding affinity assigned to peptides of different lengths? can be related to the underlying implementation of the prediction methods. So binding affinities, if we're talking about IC50 nanomolar, they are going to be directly related to the experimental measurements available. So there is no problem with the different lengths. The affinities are just if you measure them the same way for 9 mer, 10 mer, 11 mer, all these things. So that is directly incorporated in the training. For the ligand illusion kind of data where you just have yes, no, I just explained that how by using essentially an equal distribution of peptides of different lengths for the negatives and the positives, only the ones that are positive, we have a score that then learns how to distinguish these different length distributions. Jay?
Sure. Yeah. So the next question that I see is from Christopher Thorpe. Would it be possible to extend the reference allele panel / create custom sets of alleles to allow the easy specification of the most common alleles for specific ancestries, for example, from 1,000 genomes superpopulation? And so right now, we don't have an easy way to do that, but we are working on implementing a allele finder with kind of more functionality built in, where we'll have custom pre-populated sets and allow to also upload a set of alleles if it's of interest. Thanks.
Next question is, I used PepMatch on Next Generation website and noticed that it reports unreviewed proteins as well from UniProt. Is there a way I can pre-select proteins? So I think what you mean by unreviewed proteins are the Tremble entries. So we basically select the reference proteome from UniProt, which includes SwissProt and Tremble entries. And so it does a search across the entire proteome, including those. And so there's no step in the site to pre-select whether you want reviewed or not, but there is data columns that indicate whether it's from SwissProt or Tremble, and you can filter that after you download it or have the table there ready.
Next question is from Kyle Martin. Why are some alleles default and others not? What is the reason for choosing the default ones? So in most cases, the default ones are commonly found alleles in the human population. I think at this point, for class 1, there's like tens of thousands known. For class 2, with the combinatorial alpha and beta chains, it probably is millions that are possible, but are never observed. So the default is alleles that are commonly found, which then typically are more relevant in your population. Now, including all the others is still, if you are on purpose studying a certain allele, say you are interested in you have things like disease associations with rare alleles where you have a certain population that say have more diabetes and then have a certain allele and you want to examine that kind of connection. That's why it's important to provide all the alleles. But unless you're studying a rare population where that allele is frequent or a specific patient, there's typically less value in covering all the possibilities than focusing on the ones that are found in at least 1% or 0.1% of the human population.
Yep. So the next question that I see is from Mauricio. Please explain how binding and elution differ in the outcome of the prediction. Are the different sets of data sets used to develop the models? So the binding predictors will predict the strength of the interaction between a peptide and an MHC molecule, so purely just how strong those molecules can interact, whereas elution can predict whether a peptide is likely to have been eluded from a cell surface, to have undergone that whole process from processing, breakdown, transport into the ER, and make its way to the MHC binding and make its way to the cell surface. And yes, they are trained on different data sets. So the binding predictors are trained purely on MHC binding assays, so just peptide MHC in isolation, whereas the elution predictors are trained on MHC elution experiments, where you have a cell with known MHC types, and you can evaluate the content of the peptides that are eluded from the cell surface, so that you can infer that they were bound to those MHC. Thanks.
There was a clarification question. So is significance of running pep matches only related to conservancy to the proteome and not functional readout in terms of immunoactivity? Correct. So it's but because you can search a peptide at certain mismatch thresholds, so allowing for, let's say, a 15 mer up to 3 mismatches, we've seen in the literature and in our own work that there is sometimes a cross-reactivity for peptides that are not exact. So it can give you an idea based on conservation whether something could potentially be reactive.
Next question is from anonymous attendee. While there is NetMHC2Pan 4.3 EL and BA as the latest version prediction method, why IDB still uses NetMHC2Pan 4.1 EL and BA as the recommended method? That's our favorite question. It's because we are behind. So definitely for class 2, as I mentioned before, we are about to release the next versions of the next generation tools where everything is going to migrate over. So we've been delaying some of the updates on the prediction methods there. So if you're urgently needing the updates from 4.1 to 4.2 or 4.3, which are expected to be better, then you can find them on the Copenhagen website. But yes, we are actively working on this. Unfortunately, even if it should be relatively plug and play, there's always issues whenever we implement something new and there's a testing thing or so. And at some point we just had to stop development of the old legacy side as we move things to the next gen tools.
And
Thank
sorry,
you.
Bjorn, I just wanted to chime in there. So we actually do have 4.2 and 4.3 available on the legacy site.
We do.
But we just haven't updated our recommended predictor just yet, just because we haven't had a chance to evaluate. So
that's
the
what I'm saying.
recommendation, yeah. So the recommendation is still at 4.1, but we do make those other methods available.
Okay, sorry. My fault. I thought we had. Okay, so then was Discotope, I guess. Yeah, Jay is right.
Okay. Oh, sorry, I lost the question window for a second. And I don't know how to get it back. Okay, there it is. Next question is from Gonzalo Acevedo. Is the as-is option the same as peptide mode in the downloadable version of NetMHCPan? I'm not 100% familiar with the downloadable version of an NetMHCPan from DTU, but what the as-is option is if you input a list of peptides instead of full-length sequence, it will just predict the peptides as you've entered them instead of breaking them down into smaller peptides. And I imagine that's probably the same as what the peptide mode is, but I can't say for sure.
I'm not seeing any questions regarding my presentation, so I'll look for other questions that I could potentially answer. So sorry, Bjorn. I'll pass it on.
Okay, I'll take one from Gonzalo Acevedo. Sorry if I butchered the name. Do you think the difficulty predicting CD4 plus immunogenicity will be overcome with more better training data, or do you think it's biological? That's a really good question. And bioinformaticians will always say we just need more data and then we can figure it out. And I do believe that to some extent. But I also think there's going to be other ways. I mean, there are going to be some things, not just more data of the same, but also understanding more once we have more paired. This is the CD4 T cell receptor, and this is the peptide and the MHC to kind of get the trimer right. I think that's going to be one of the things that is currently missing, essentially, like the ability of finding TCRs that are going to be recognizing something. That should be at least a factor of 2 if we get that better. Yeah, and then there's other things, such as say, for example, did we already talk about this? I don't think so. Peptide expression levels, say. So some of the things are just variables that are outside of it right now. So a peptide might have all the features that are necessary for recognition, and you say, well, this is a peptide found in TB, but it's never recognized because the peptide is actually not is in a protein that is not expressed. So there's like a trivial reason. So essentially, we are not expecting ever to have 100% accuracy if we just go by the peptide sequence and the MHC to predict immunogenicity. The context in where this happens is going to be critical, and essentially, depending on the context, other factors might be necessary that we can't put in a general prediction.
Next question is from anonymous attendee. What is the binding prediction based on? Is that prediction going to be updated with new deposited data? So the binding predictions are based on MHC peptide binding data sets. So experimental data sets that are generally deposited into the IADB. There are some private data sets that that are used as well. And we're working to try to overcome that and make most of these data sets public. And yes, are they going to be updated? We'd like to try to reevaluate whether or not there's enough new data to make retraining worthwhile. And we recently assembled a new data set, and it looks like we have a lot of new data available since the last time that we retrained. And so I'm expecting to see some updated models soon-ish. I don't want to say exactly when.
Next question I have is from Maria, who asks, when 50-mers are used for MHC class 2 epitope prediction, core 9-mer is the same for multiple 50-mers, however, the percentile rank is different, even if core 9-mer is the same. Why is that? Does it mean if the core peptide is shifted, it is less likely to be presented? So I think I probably overemphasized that it's just the core that binds. Essentially, when we are adding the flanking residues, we do find a little bit of specificity and a little preference for having like where are the other, so there's contacts made kind of like to the backbone of the peptide, and where those are can aid to the likelihood of the peptide being presented. So it's not just the core that makes the prediction, determines the affinity. There's these other contributions, but they are like in the maybe less than 10% of the overall effect range. So I kind of like skipped over that, but you're completely right. There can be some differences, but they should be quite minor.
So next question I see is from Ramona Carr. What's the significance of the median percentile and peptide epitope prediction? So the median percentile that we report is simply if you select multiple binding methods, each of those methods are going to report a percentile rank. And the median percentile is literally just the median of all of those percentile ranks for that peptide among the various methods that were selected. And I think we actually report median percentile even if there's one, but that's just going to be an echo of the percentile rank for that one binding method. Thanks.
This is from Anonymous. So for detecting similar peptides, would you recommend PepMatch or BLAST or both? So I guess it depends on what you mean. So if you mean like if you have a list of peptides and you want to see which ones are similar, then we recommend the cluster tool. So that will cluster peptides that are similar together based on sequence identity. But if you want to find similar peptides in other species and their proteomes, we recommend BLAST for short sequences, or sorry, PepMatch instead of BLAST for short sequences, and BLAST would be for more protein alignment. We show in our PepMatch paper that PepMatch is deterministic, so it can find every possible match for a given threshold, way more than BLAST can and other similar alignment tools. Again, to stress that it's for shorter sequences.
Next question I have is from Saurabh Badwal. How would we rank 7 allele versus 26 promiscuous allele? So we actually just made a comparison recently again, and so far they're quite even, which is still a surprise to me. The 7 allele method was developed 2015. I think, as you saw there was a bias towards DRB alleles. Back then, also the predictions for DP and DQ weren't that great. I was hoping that we would now be better. with using a broader panel of alleles, and I would be immunologically assuming that they should be better. But so far, we haven't uh, found that. But I think there's room for systematically analyzing that again, and maybe there's some mixture in between, or maybe we can expand the 7 to 11 or so. But right now, I mean, in our own work, we've stuck to the 7 allele approach. It's somewhat nice to be conservative and stick to something over and over, because then it's easier to, you build another large data set that is generated in a similar way, and then you can show more consistently if we can move on from there. Jay?
Yep. Next question is from Anonymous. If one predicts the binding, wouldn't that imply that the peptides were processed? Thus, do you see positive correlation between these two? So, great question. And no, if you predict binding, so the binding predictors again are trained simply on a data set of MHC molecules and peptides in vitro. Do those molecules, will that peptide, it can be a synthetic, it's usually a synthetically generated peptide, will that peptide bind to MHC without taking anything else into account, no surrounding sequence context, just the peptide and the MHC molecule. The elution predictions will take into account processing. So if you have a positive result from the elution predictions, that indicates that the peptide has kind of gone through that whole pathway of it was processed, it was transported to the ER, it bound MHC, it made its way to the cell surface. So that's where that indicates that the peptide was naturally processed.
I believe I can answer this one
Dan,
based on peptide structure. So as recent publications point out that peptide structure is more relevant to binding prediction than sequence, which explains in some cases why one might have different peptide from the selected one, yet binds and gets recognized by a different antibody. And any thoughts on that? So yes, we have collaborators in Denmark that are working on this. So like the methods we have now are sequence based, but hypothetically, if you have the structure as a part of the predictor or as your input into a neural network, it would be much more accurate hypothetically. And so yeah, our collaborators are working on structure-based prediction that potentially could be better, but nothing as of right now that's in production.
Next question I have is from an anonymous attendee. Hi, my question is whether analyzing human versus mouse data affects the choice of analysis tools at any step of neoantigen discovery, e.g. binding affinity prediction, etc. So of the tools that we're currently recommending, we like the NetMHC, PAN, whatever suite of tools for both binding affinities or ligand elution likelihoods, they are trained on both, and they are actually trained on including some other species as well. And we would also recommend them for definitely for mouse. The last set of tools that was really clearly a difference was the Tap molecule transport efficacy, which is very different from, or actually quite different, not very different, between humans and mice. But that's a good example for where in the ligand elution predictions, the effect of the difference in the Tap molecules is essentially reflected by the peptides that are found on the cell surface. So that's why these tools tend to work also for mouse versus human. Now, if you go beyond human and mouse, very often we have not enough data sets to be really, really confident about these, and we can't say this for certain, but at this point, we would still recommend the same set of tools. Jay?
Yeah, so next question I see is from Myriga Saad. I need a clear explanation on peptide index and peptide percentile rank. So those are two very different values that we return in the T-cell class 1 tool. Peptide index is simply a unique identifier, integer identifier that we assign to a peptide in the result set. So if you have a protein sequence that is 1,000 residues long and gets chopped up into 1,000 peptides, you're going to have a peptide index from 1 to 1,000. The percentile rank is, we generate percentile ranks of the binding affinities or of the elution scores, and the percentile rank indicates where in a distribution of randomly selected peptides, if we just look at random peptides, we look at the distribution of predicted values, where in that distribution does the IC50 value of this peptide lie? If it's at the 1 percentile, it means that it binds better than 99% of background peptides. And so, yeah, it's clear from all the questions on percentile rank, it's clear we need to do a better job of explaining that on our website, and we'll definitely update the documentation. Thanks.
There's a question again about BLAST and PEPMATCH. So BLAST also has optimized parameters for short sequences. Did PEPMATCH surpass BLAST in that mode too? Yes, so I'll link the paper in the chat. We did 4 different benchmarks for various different applications for like searching exact matches and mismatches and neoepitopes versus or viral epitopes. And not only was PEPMATCH able to find every possible match, whereas BLAST could only find 70 to 80% of the short sequences, it's also PEPMATCH is much faster based on our benchmarking. So we do recommend that for short sequences.
So I'm going to summarize a few questions that came up and I'm just going to read one part of one. Is your allele panel with 97% coverage purely theoretical, or do you have published data to support its use? I think we need to be precise what we mean by that coverage. It means that if you take random people all over the world, 97% of these will have one of these 27 alleles. That's what this means. It does not mean that you're going to have all of these epitopes recognized in all of these alleles. It just means that if you analyze these 27 alleles and you have a peptide epitope set that has candidates for each of these alleles, you're going to have hits all across the world, right? That's what this 97% coverage means. It doesn't guarantee anything in terms of will these actually work or so. It's more, so this is not theoretical at all. It's very much experimentally shown based on data on HLA frequencies across the world. But again, I think coverage is probably a tricky word here. That's it, Jay?
Yep, and then related to that, there's a question from Evans, Malin Log Tong. Are the 27 alleles of human source? Yes, yeah, the 27 allele panel is all human.
So if you don't have anything, Dan, I'm scrolling through here. Obviously, we still have a lot of questions left over. Some of these are a little hard to understand, so we're focusing on the ones that we immediately understand and can answer. If you don't answer a question, maybe we're also out of time now anyway, and I'm really tired. Maybe Jay can do a last one. But otherwise, I mean, very much we have the help system online. Please go there and post them again. In some cases, we're just lacking context to some of these questions, and it would be easier to then go back and forth in like a help ticket or so to explain these more. We don't want to neglect any of them. This format makes it a little hard to have this as a dialogue, obviously. So Jay, you want to do a last one or so?
Yeah, I think that's everything I feel comfortable answering right now.
Okay.
Awesome. Thank you so much, Jay, Dan, Bjorn. That was an extra mammoth long Q&A session. So thank you all so much. And that brings us to 10:20 a.m. US Pacific time, which is officially the end of our first section, which is a bit longer than yesterday's first section. But thank you all. Thank you all for being so engaged and sending through so many questions. And apologies we couldn't get to them. So what we'll do is either, respond to a few written, so check the answered column, or as Bjorn mentioned, if you could please email us at help@iedb.org. I will pop that in the chat. and we will engage that way. And as Bjorn said, it's just easier to go back and forth with a little bit more context. So thank you all for your understanding. If your question, we didn't get to it or we didn't get to answer it, or perhaps there was just a little bit of context missing. So yes, much appreciated. And with that, we'll start our 15-minute break, well deserved for everyone. And I will post some resources in the chat again, the timing, and yeah, we'll see you in 15 minutes at 10:35 US time. Thanks all.